Assalamu alaikum dear students, welcome to your channel The Anatomy Canvas. In today's class of Embryology Foundation module, we are going to discuss with the process of neurulation. So learning objective for the today's class are to discuss the process of the neurulation, to describe neural crest formation, to discuss their clinical correlates, and to discuss the ectodermal germ layer derivatives. So embryonic period is the period of organogenesis that occurs from 3rd to 8th week of development, in which all the three germ layers give rise to a number of the specific tissues and organs. And this is again another MCQ for your exam, that the period of embryogenesis or organogenesis is from 3rd to 8th week of development. So now move on to development of the central nervous system and special sense organs. There are the three different sources from which CNS and special sense organs originate. These are the first source, second and third. So the first source is the neural plate, and neural plate gives rise to the central nervous system, the somatic motor nerves and the preganglionic autonomic nerves. Second source is neural crest cells, which remove themselves by epithelial to mesenchymal transition from the plate and migrate away just prior to its fusion into the neural tube. So the neurons and the ganglia of the peripheral nervous system, that is somatic, sensory, somatic and autonomic ganglia, post-ganglionic autonomic nerves and suprarenal and chromaffin cells. So these all are derived from the neural crest cells. And third source for development is the ectodermal placodes. These ectodermal placodes contribute cells to the cranial sensory neurons and form the olfactory epithelia, the epithelia of the inner ear, and by a non-neuronal contribution, the lens of the eye. So these are the different sources from which the central nervous system and the special sense organs originate. Now we discuss all these in detail. So what is the process of neurulation? The process involved in formation of neural plate, then neural folds and closure of the folds to form the neural tube is called neurulation. So this is the definition. You must memorize this definition, that it is formation of first neural plate, then neural fold formation, and when these fold close and form a closed tube called neural tube, the process is complete and it is called neurulation. So neurulation starts when, as a notochord develops, it has an inductive influence on overlying ectoderm and thickens and form the elongated neural plate. So in this diagram you can see, at this stage of development, it is still a broader disc shape. The embryo is in a disc shape, which is more broader in cephalic direction than in caudal direction. Here is the region where you can see the primitive pit is present, and this is the region from where you can see the precaudal plate is present. This is the region of the connecting stalk. So look at this diagram. This section B is taken at this level, and in this diagram you can see in blue color this is the ectoderm, and in red color you can see the formation of the notochord, which is in process of development, and it is in different stages which we have discussed in the previous lecture. So as the notochord is forming, it also has an inductive influence on the overlying ectoderm to form the neuroectoderm. So as it is forming and it becomes a tubular structure, the notochord become tubular structure, and as well as the paraxial mesoderm is formed over here. So this neuroectoderm also forms the neural folds and neural grooves. So origin of the nervous system, again you can see nervous system develops from neural plate. Neural plate is thickened area of the embryonic ectoderm. Here you can see this is ectoderm, which overlies the notochord, and this notochord has inductive influence on ectoderm, and this ectoderm becomes the neuroectoderm. So neural plate then differentiate to form neural tube, neural folds and neural crest. So its edges elongate and they becomes the neural fold, and depressed area in the center form a depression called the neural groove. When these edges coalesce and they fuses, they form a closed tubular structure which is called the neural tube. So neural tube differentiates into the central nervous system, that is the brain and the spinal cord. Also, neural crest cells are formed from it, and they will give rise to the cells that form the peripheral and autonomic nervous system. So neurulation is the process it appears rostral to the primitive node and dorsal to the notochord and mesoderm adjacent to it. So please understand, this is rostral to primitive node. At this area you can see this is the primitive node and it appears rostral or cranial to primitive node. So when you are looking at the surface, when it is a broad shaped structure, you can see this is the area which will form the neuroectoderm, and in cross section you can see this is the area which is present dorsal to the notochord. So this ectoderm which is dorsal to the notochord and it is present cranial to the primitive node, this will form the neuroectoderm and it will form the neural plate. This neural plate broadens and extends cranially as far as oropharyngeal membrane. So here you can see in this area oropharyngeal membrane will be present and this neural plate will extend up to the oropharyngeal membrane. Fusion of the neural tube starts with the 4 to 6 somite stage at the level of the somite 1 and 2. Usually in the cervical region, first fusion starts and usually this fusion occurs in the region of the 5th somite. That is, in general it is 4 to 6 somite, and here fusion starts, then this fusion moves both cranially and caudally. In this diagram, you can see the different factors which are involved in molecular regulation of the brain development. So in this diagram, you can see this is the notochord and this is the ectoderm and different factors which are responsible. So this notochord release factors caudin, noggin and follistatin. These three factors are inhibitors of the BMP. The BMP is bone morphogenetic protein. So remember, this BMP is present in higher concentration in the ectoderm and is responsible for ventralizing the ectoderm, and it because of the ventralization of the ectoderm it will form the mesoderm. When this BMP is inhibited by the factors the caudin, the noggin and the follistatin, and also the fibroblast growth factor is also involved. So inhibition of BMP leads to upregulation of fibroblast growth factor, which will lead to formation of the neuroectoderm. So there is the interplay of the different factors which is responsible for brain development. So please understand this point, that in the ectoderm normally BMP is present, BMP4 and BMP7 both are present in the ectoderm, and these BMP are responsible for ventralizing the ectoderm. So when these BMP are inhibited by different factors, that is the fibroblast growth factor which induces caudin, noggin and the follistatin, and these three factors inhibit the BMP. As a result of inhibition of BMP, the ectoderm will be dorsalized and it will form the neural plate. Also remember that these three factors are responsible for dorsalizing the neuroectoderm and formation of the forebrain and the midbrain structure. For the formation of the hindbrain and spinal cord, the WNT3A and fibroblast growth factors are responsible, which will then depress the BMP, inhibit the DMP, and then the dorsal structures of the central nervous system, that is hindbrain and the spinal cord, will be formed. So there is the interplay of the structure between these different factors that is responsible either formation of the brain development, or when BMP is present in higher concentration, it will lead to ventralizing the ectoderm. So now look at this diagram in which you can see the fusion, the process of the fusion has been started, and it progressed both in cranial as well as in caudal direction. Here you can see this is the anterior neuropore, and here you can see the posterior neuropore which is present, and through these neuropore initially this closed neural tube is in contact with the amniotic cavity. Later on, these neuropores also closes. So the anterior neuropore usually closes at the stage of the 18 to 20 somite at day 25, and the posterior neuropore closes at day 28 when it is a 25 stage somite. So in this stage you can see this is a 28-day embryo in which you can see both the anterior and the posterior neuropore are closed, and the neural tube is present in the form of a closed structure. Also you can notice at this stage the lens play cord is present, the otic play cord is present, also the first, second and third pharyngeal arches are present. At day 28 you can see 3 pharyngeal arches, at day 25 you can see 2 pharyngeal arches. Also this is the area where pericardial bulge is forming, where you can see the heart bulge, the pericardial bulge in which heart is developing. This is the area of the connecting stalk in which you can see the vitelline duct, the umbilical vesicles, umbilical cord and allantois. This is the area you can see where the limb ridge is present. So up till now we have studied that the notochord basically has an inductive influence on the ectoderm to form the neuroectoderm. So this neuroectoderm, then its edges elevate and they form the neural tube and neural folds, and when these folds closes in the region of 4 to 6 somite and their closure progress in both the direction, they will form a closed tube called the neural tube. So in this diagram also you can see the closure is in process. In the center it has been closed, and here anterior neuropore is present as well as posterior neuropore is present. In this diagram you can see the pharyngeal arches are present. This is the mandibular prominence, the maxillary prominence. Here you can see the anterior neuropore is present. Here you can see this is the somite one, and we can count the somite from here to this area, and this is the stage of 15 somite, 14 to 15, when still both the neuropore are open. So again you can see in this diagram slowly this is a broad disc shaped structure when embryo is in the form of a flattened disc. Here you can see Hansen's node or primitive node. This is the primitive streak, and the ectoderm in this region which is cranial to the Hansen's node will form the neuroectoderm. So here you can see the elevated edges of the neuroectoderm which are forming neural folds, and depressed area in the center forming neural groove. When these folds fuses with each other in the region of the 4 to 6 somite, they starts to forming a neural tube, and this fusion then progress in both cranial direction as well as caudal direction. In this you can also see this is the broadened area which is forming the brain vesicle, and this part will form the spinal cord, area of the spinal cord. So now we move on to the development of the neural crest cells. That is very important, and different factors which are also involved in the neural crest cells. So neural crest cells are formed at the dawn of the somite stage when still neural folds are present but the neural tube has not closed itself. So at the edges of the neuroectoderm, the cells which are present over here, they show epithelial to mesenchymal transition. They dissociate from this neural plate and come to lie on sides of the neural plate, and they form the neural crest cells. So please also remember there is again an interplay of the factors over here. That is the BMP and also the fibroblast growth factor. So BMP is present in higher concentration in the ectoderm, which is responsible for ventralizing the ectoderm, and in the region of the neuroectoderm BMP is inhibited by fibroblast growth factor, which through follistatin, caudin and nogin inhibit the BMP4, and this area of the ectoderm will form the neuroectoderm. At the edges of the neuroectoderm, the area which the cells which are destined to form the neural crest cell, there is a moderate concentration of the BMP4 and 7, and this area will form the neural crest cells. So in this diagram you can see the closed neural tube, the neural crest cell, and surface defect in epithelium. Ectoderm has been completed, closed and complete ectoderm is present over here. Also you can see the notochord which is present over here. So now when the cells dissociate from the neuroectoderm and they migrate in different areas, so they then have the two pathways. The one is the dorsal pathway. Through the dorsal pathway, especially in cervical region, in the trunk region, the cells migrate into the surface ectoderm where they will form the melanocytes. And the ventral pathway, they move cranially and ventrally and they will form the dorsal root ganglia, then the autonomic ganglia. Also you can see over here the this is the cells forming the chromaffin cells in the adrenal medulla. Also they are forming the enteric nervous system and populate the GIT. So these are the different pathway, the dorsal pathway and the ventral pathway of the neural crest cells where they migrate in different direction. And this is the area of the trunk. In the area in the cervical region, through the dorsal pathway, these neural crest cells then form the craniofacial mesenchyme, which will lead to craniofacial structures. So again you can see the different cells, the neural crest cells, they are forming the melanocytes, then they are forming cells of the dorsal root ganglia and ganglia of the cranial nerve. Then the cells, they are forming the autonomic ganglia, then the cells forming the adrenal medulla and also the Schwann cells. So here you can see this is a closed neural tube and this is the notochord. In this diagram, you can see the two pathway. The one is the ventral pathway and one is the dorsal pathway and two is the ventral pathway. So through the dorsal pathway, they enter the epidermis into the surface ectoderm where they will form the melanocytes. And through ventral pathway, they will form the different structure, that is the dorsal root ganglia, the autonomic ganglia, the chromaffin cells, the Schwann cells, and the splanchnic nervous system of the GIT. So in this diagram, you can see the neural crest cells derivatives, that is they form the connective tissue and bone of the face and skull. So please remember, each and every line of this slide is your MCQ. So you have to learn, you have to memorize this slide. The neural crest derivatives, first the connective tissue and bone of the face and skull, then the cranial nerve ganglia, the C cells of the thyroid, again very important MCQ, the conotruncal septum in the heart, again a very important MCQ, the odontoblast in teeth, dermis in face and neck, the somatic sensory and postganglionic nerves, the somatic and autonomic ganglia, adrenal and chromaffin cells. So you can see these neural crest cells are forming a large number of structures. So sometimes some embryologists consider these neural crest cells as a fourth germ layer as well. So please memorize these, all the derivatives of the neural crest cells. Another very important slide, that is the ectodermal germ layer derivatives. So that is the central nervous system, the peripheral nervous system, the sensory epithelium of ear, nose and eye, epidermis including the hairs and nails, the subcutaneous glands, mammary gland, pituitary gland and enamel of teeth. So please remember, these all derivatives are also very important from your MCQ's point of view. So again another slide, each and every line of this slide is your MCQ. So please also memorize this slide as well. Then we move on to the neural tube defects. So you can see there, if the cranial neuropore do not closes, this will leads to condition called anencephaly, in which the cranial neuropore or anterior neuropore is open and the brain tissue is exposed to corrosive effect of the amniotic fluid. This condition is diagnosed in prenatal life and pregnancy is terminated. It may be if the there is a failure of the neural tube to close caudal to the cervical region, then condition is called the spina bifida. The spina bifida also, the condition varies with the severity of the condition, whether the spinal cord and its nerve roots emerge through the defect or only it consists of the meninges. This is the condition in which although the neuropore is not open like this, but through a defect the brain tissue along with the meninges protrude through the defect and they form the encephalocele. So this is the picture of a baby having the anencephaly, in which you can see the structures of the brain are exposed and they are damaged. This is the picture in which you can see the spina bifida. So please also remember, there is another MCQ over here, that most common site of the spina bifida is the lumbosacral region. Here usually this may be the area which is more vulnerable to environmental or genetic factor. As a result, most common place where the spina bifida occur is over here. So this is the slide where you can see the spina bifida. So here I complete my today's class. The references are Langman Medical Embryology and Developing Human by the Keith Elmore. Thank you very much for your attention, dear students, and Allah Hafiz.